Okay, well, thanks to our panellists for sitting up here. I think it's always a really good sign when you've got more people in the audience than you do sitting up on the panel. Always a bit awkward when that happens. Anyway, so I've got a few issues that I've been writing down through the day that I'd be really happy to bring up with the panel to discuss and everyone to comment on as they see fit. But firstly, are there any questions from the audience in the room which they'd like to direct to the panel on anything related to microsampling, noting that the theme of this is really designing microsampling studies and building collaborative networks, but really anything related would be good. So we've got Michelle Cree here.
My question's more, we're not probably there yet, but it's like, how far are we away from microsampling of blood cultures?
Microbiology. We asked the infectious diseases physician.
Well, in terms of yield from blood cultures, the critical component, as far as we know, is volume. And well, that's right. But really, the kind of volumes we're talking about are so many orders of magnitude different that it's not foreseeable in the near future. And the multiplex PCRs even require a decent volume, I think, from blood, and their accuracy and sensitivity is not as good as blood culture. So at the moment we're sort of stuck for blood cultures with a minimum of 5 to 10 mils per bottle, and that's what it's going to be for the next little while.
I think until we can bypass the culturing step, that's where we're going to be. Any other questions from the audience? Okay, my first question to the panel is regarding the training that's required to perform microsampling in studies, and how important, obviously the answer is going to be yes, but what level of training is required to make sure the correct procedure is followed and that the timings of the sampling are done correctly to ensure that the results are as valid as possible. I'm actually going to look towards Brian and Lawrence for this because I think that you've got very good experience with this.
Oh, I mean, in terms of our Medang studies, we're really only just starting to implement and get ready to put microsampling into practice. But I think given our experience in Port Moresby with the collection and the quality of the samples that have been collected, we've been working with other colleagues to put training videos together. We're actually going to spend quite a lot of time training the nurses in how to, the best site for pricking the finger or heel to get a good quality blood spot. And then, of course, going right through into drying times and having to store it correctly. So a lot of training, I think, is going to be required in order to get the quality of samples that we need, especially from neonates and those sort of special cases. So I guess we're learning as well through the process of what is necessary and what is best practice. And there's a bit of trial and error at the moment, but education I think is the key to a successful study for this.
So whilst other people are thinking about this, I think for a lot of the issues that I'm going to raise, I think that there's a really strong argument for trying to have some early opinion-based publications on what these are, to try and share the experiences that you've had with the unique studies that you've done, how you've tried to solve those problems, such that if other people who also want to follow suit, then they've got a bit of a guide for how they can shortcut through to a better solution. So I think that that's a really good example of a short communication that could go into a relevant journal to say, this is how we have addressed this problem. So I think that's useful. Does anyone else want to comment on that issue?
In relation to the timing, the key thing is actually not so much the timing of the sampling, it's actually that they record the time they take the sample. And so we've been very rigorous in telling the nurses to not worry too much if they're 10 minutes late, but not to record it as what they should have done rather than what they did do.
Okay, so the next question is relates to future use of microsampling, and this one's going to go to Susie in the first instance, about whether or not microsampling, because it is perhaps more feasible than having to take a venipuncture to sample, does this mean that we should be aiming to do larger studies, making the most of the population pharmacokinetic modelling, which Sam discussed in detail what the advantages of, so that we can actually have much more statistical rigour in our models and therefore better empiric dosing regimens that are derived from that? The answer is yes, but I'll let you go.
Yes. Yeah, I do think that, and if we can do larger studies, even with our bridging studies, a lot of the data that we now compare, where we're looking at special patient groups, there's a lot of data that we've got now in the literature to compare to, but these bridging studies, there's very little information. So if we can have a larger, more powered study, then we'll have a better baseline.
But in due course, do you think that we can, if it is that we can replace the traditional sampling, that our approach actually should be to aim to recruit more patients? Because hopefully it becomes more feasible to do those studies.
Yeah, absolutely. Yeah.
Does anyone else want to comment on that?
I've also a question, Jason. With all the
Please use the microphone, Brett.
Sorry. Oh, sorry. Sorry about that. The question I was wondering is, we're using microsampling for all these studies, but what happens if it rolls into routine service? So as a routine TDM service, say for pathology then, and all I can hope, Steve, is that you've got all these problems fixed up and all the regulations done before it rolls into pathology, because, it's good to get to that point, but it's then have to go into routine practice. So what's going to happen then, I wonder? So that's what I'm saying.
This is actually, your area of expertise.
Well, I'm actually putting the question
answer,
back.
but I think you'll correct
I'm putting
me.
the question back to the audience. I'm saying, what do you think is going to happen then? We're getting to a point now. Then what happens when it goes to routine service, routine TDM service?
I was actually sorry. This is answering Brett's question, I think. But we were talking about regulations before and what you needed, and there was no prescription. But in some ways, for the translation to occur, wouldn't it be really good to look at what the lab's regulations are in terms of method validation? Because then that would make a more seamless transition in the long run.
Well, that's right. I think you've got to end up where we're going to end up being is what are the regulations for that, and that's your goal, because you can your own, but if it doesn't match what's in the therapeutic lab or the pathology lab, but then you have issues again, there's more work. So I think the goal is know where you want to go to. It's perhaps from here to there, you've got to work your way. And we've got NATA and everything else to consider, and this is going to be something new for NATA to look at in the future as well.
So on that issue, I wanted to ask about the acceptance criteria, just sort of exploring a bit more on the question that I asked Steve before, and whether or not, if it does make it into clinical use, that we should we be accepting values such as a 15% level of error in some of our analyses, or should we be aiming for better if, from a laboratory perspective?
From my perspective, much better, because on top of that, you've got uncertainty of measurement on top of this as well. So a clinician will want to know a value is X, and you've now given me a value of Y. Is that different? And it's not just the analytical variation within the laboratory, it's intra-patient variability, the whole lot, and all of them are cumulative. So if you can reduce your CV or your precision, tighten your precision off your assay, it's going to make that a lot better. And that's going to be the major impact on your uncertainty of measurement. So for us, from pathology side, they're always asking what's the uncertainty of measurement for each assay.
So what sort of percentage value do you think you might be trying to aim for, obviously noting you don't know?
Well, I would say for most of our inter-run, 7 to 8% tops. If we can do better, we can do better. That's you want to be under 10% easy.
Stephen, did you want to comment on that from a research laboratory perspective?
Yeah, when it comes to acceptance criteria, I do remember when the FDA said 20% was okay, which meant that 2/3 of your values could be outside of 20%. If just using the QCs, you had to get 4 out of 6 right. But in our lab, if the criteria are 15%, if it's sitting above 10%, we're really worried because it's not going to stand up very well. When we validate, if it's within 5%, we're really happy with that, and we might have, if you've got 3 different levels, one of them, precision or accuracy, might be a little bit higher than 5%. Happy to go with that.
So if anyone's got a question, please put your hand up. Oh. Okay, Rhonda?
I think maybe with the dry blood spots, it may need to be a fraction higher because we need to take into account variations in the spot punches as well. And so I've just worked on like a 10% and calculating that through.
I think from a pathology point of view, I can understand that you can see the advantage of the microsampling, but it's going to have the same precision because otherwise you're giving on one hand and taking on the other. And so it's like the old ratchet effect. We're here, we want to improve, we don't want to give up that and come back to where we are. What have we achieved? We want to make sure we're here, we're improving, we're going to here now. And so I think it's going to have to match at least the same precision as the liquid base, and then you've got the advantage of the small sample volume.
Oh, sorry, Darren. Regarding the future with different formats and things like that, you do have the miniaturization. You can stay with a liquid plasma sample, but just scale everything down. And as your machines get more and more sensitive, we can achieve microsampling without a great leap in practice. But I guess then, if you wanted to look at a different format where you're putting the sample onto a substrate, that has to be compatible with your instrumentation and the goal of getting a lot of stuff through fast with the right answer.
As a non-laboratory person, I'm just wondering, are we at the stage yet where we know enough about the limitations of the different alternative methods of sample collecting relative to the drug? So for example, we know that some drugs stick to plastics. We know that if we've got a small volume of blood and we put that into a gel tube, that we increase the error on the drug measurement. Just wondering if we know enough about, for example, the relationship between physicochemical properties of the drugs and if it's likely to stick to the different components of either the VAMs or the capillary or the dry blood spot paper. Like, for example, Lawrence made the comment about penicillin, I think it was, sticking to the plastic in the PICC line. I think someone said something about the naphtha drug and that was sticking with the... I don't know. Are we at that stage yet? I'm just thinking about the work I work in, pesticides, for example, and they tend to be very lipophilic and stick to lots of things. Just wondering if we could use these devices for my research or we don't know.
Thanks, Darren. We don't know across the board. So it's part of the laboratory-based validation where you need to make those assessments. And I think if you have seen, just at that starting stage of study design, is if you've seen anything in the literature where you see something sticking, I think you just got to look and make sure you've had a thorough look on your way through before you take it into anyone.
Hopefully part of your method development up front is if you pick the drug as your target, you want to know what stability is like. You've got to work, and if it's unstable, you've got to work within those confines as well. Hopefully you know if it's pH dependent, all those properties about it. So you've got to know as much as you can about the target, and you've got to work within the constraints because it dictates to you, and you've got to work with that. So again, if it's very lipophilic and it's into plastic, well, you can't use plastic. If you're using glass, sometimes you have to have siliconized glass. So they're the constraints you've got to work with, and you can't change some of those constraints. That's what it's got to be. That's what it's got to be.
It's a good question. I think a lot of it depends on the amount of studies that have been done with drugs and then trying to perform simple mathematical regression analyses to look at whether or not any of those factors are associated with an impaired ability of that microsampling device, or the inaccuracy associated with it. So I think it's a really important.
The other thing too can happen is that sometimes manufacturers change things and they don't tell you, and then suddenly your assay just falls out of specs and you don't know what it is, and then you've got to go back and try to find it. And the manufacturers, no, we haven't changed anything, and finally, oh yes, well look, we've used a different gel now, or we've got this or whatever else, but it's taken a long time and hard work to find out what the issue is. So sometimes some of those things are taken out of your hands. You just got to have checks and balances in place to pick it up early, and then how are you going to go about addressing it?
Dave, Darren, another aspect to your question, which format do you follow? In our lab, we've tried looking at dried blood spots, dried plasma spots, plasma VAMs, whole blood VAMs. We've got some Novoplex cards we haven't even opened yet, because the burden to run all these validation tests against all these different specimen types, it can slow the whole process down, and sometimes you better just pick the format that matches your application and then just make it work. Hopefully it will work for the one you're interested in. Otherwise you have to change.
Just to sort of add to that, I think the example that Susie gave earlier about the binding that the vancomycin seemed to have to the filter paper occurred with drying at 22 hours, and I think what we should be doing is making sure that our sample handling is consistent. So in the clinical studies, obviously it's a really interesting observation, but in the clinical studies we should be making sure that the drying, there's a standardised drying time of an hour, and the samples then get put in with the desiccant, and so it's all done properly, and that allows you to minimise some of those factors, like time, sound like some kind of time-dependent binding to the card. So, and those things are completely feasible, certainly in the current clinical environment.
Okay, we've spent a lot of time talking about analysis of the microsample. I wanted to ask a question to Sam, because he's in the audience, about analysis of data associated with microsampling, and how much help can the different pharmacokinetic analysis approaches, specifically population pharmacokinetics, address some of the problems that are maybe inherent at the moment to microsampling. So the system errors that are introduced by microsampling, how much can the method of analysis minimise the potential longer term problems with use of that data?
Thanks for the question. So at the moment, we are doing studies with dried blood spots, but we're yet to do a clinical study which has looked at their PK. In the validation studies, we have looked at the PK. And I guess you can't get rid of error. It's there, it's inherent, it will be tied up in that residual unexplained variability. And the more residual unexplained variability you have, the less likely you're at risk of not finding relationships. But we were discussing earlier that often the types of relationships that we're interested, at least in when we're doing these drug trials, are large, clinically significant relationships. For example, in our pregnancy studies, if the clearance rate is more than 50%, even if there was a 15% CV as opposed to a 10% CV, we think we'd still pick that up. But certainly you increase your risk of missing those perhaps more subtle relationships, and no amount of fancy equations and non-mem is going to fix that problem.
So I've got a follow-on question, which is just escaping me at the moment, so I'm not going to ask it just yet. That's right. I remember what it was. So the question was about when you're doing a multi-centre pharmacokinetic studies, sometimes the way that one site is performing their element of data collection and sample collection can be different to the way that others have been if maybe there's not a standard operating procedure in and around that. And do you think that that's actually something whereby modelling can be very useful, that if you look for the covariate of the institution and find that maybe one institution is associated with the data with that with much larger error, that you can predict that maybe that there is some issue with that institution and go and find out how they did their collection and use that as a way to work out whether or not you include or exclude some samples because of a systematic issue?
That's an interesting question. So I always start modelling by looking at the actual data, because I think that's the most valuable, and often you know the kinds of relationships, or you can gain lots of information just by looking. And if there is poor quality data, it's normally in that data checking stage visible. If it's very feasible, and within the program that I use, NOMEM, it's and probably others as well, very flexible in allowing not necessarily different sites having different covariate values, but different residual unexplained variability values. I can't say that I've had any personal experience with that, but certainly if one site had 20%, another site was nearing 50 or 60%, you can try to build it into the model, but ultimately it may be a matter of having that data there and then not there, and a lot of this, as I mentioned before, a lot of the modelling isn't just banging it in and getting the answer, it's making sure that the answer you're getting makes sense and you've considered all possibilities. So be in that evaluation phase, I'd imagine.
Great answer. Okay, so the next question I had for the panel is about even less invasive microsampling techniques. So instead of looking for blood, and this is extending upon what Yamali spoke about in her presentation, what about non-blood matrices? And I'm alluding specifically to saliva, sweat, and other things which are more readily available and less invasive. When do you think, are we at a stage whereby we're actually using that? I know we are for some drugs.
In pathology, we actually routinely measure salivary cortisol for diagnosis and for people with Cushing's. We are also looking at some of the other things for saliva as well. So it is a big interest to us at the moment. We've done salivary mycophenolate and comparing it to free mycophenolate in AUC studies. So over the last 4 or 5 years, the only issue I suppose you have with saliva is that you are looking at free portion. So something like mycophenolate, which is highly protein bound, about 90%, levels are going to be lower in plasma, in saliva. So again, there's another issue. You've got to low levels. You're going to have good sensitivity. And I suppose you've got to look at the correlation between what you're finding in saliva and how it correlates with plasma. And I suppose in the free levels in plasma as well. So, and is it affected by saliva flow? What do you collect it into? The container? Do you do the salivette, or do you do the drill method, where you just drill it into a container? If it's highly lipophilic, does it stick to the container? So there's all those things that you want to consider.
So is that almost another parallel area of bioanalysis which is existing with microsampling, whereby you have all of these issues in that specific space and they all need to be addressed? Or do you think that it's something whereby we should be trying to bring them together and be thinking about a suite of possibilities for sampling and then applying them? And
I think it's horses for courses. What is the best way to tackle that situation? As I said, with saliva, people don't mind doing it to a large degree, but the problem is if you have a paediatric sample, you can't get a saliva sample for it. So it's not an option that way. But for other things, it's quite easy to have, and so people don't mind doing that. So unless you're the analyst at the other end who's got to work on saliva, yeah. But then there's issues with saliva too. So if people have bleeding gums or whatever else, you can get contamination that way.
Something that Rhonda mentioned earlier with the omics and metabolomics and things like that. Looking at one of my favourite websites, the Bioanalysis Zone, every now and again you see an article about, or some sort of post, where they're using, kind of like, dogs can smell when people are getting sick and all that sort of stuff. Some sort of electronic nose where they're picking up the volatile compounds in breath that might be a, some sort of metabolite or whatever it is of a disease state. Maybe not. Maybe there's some sort of, I don't know if you can do pharmacokinetics on that, but there might be a future there. That kind of
Using it
stuff.
for diagnostics, aren't they? So
Yeah, better than, say, taking a photograph of somebody's aura and measuring stuff.
You've actually got a PhD student in Brisbane who is a GP who's working on sweat at the moment and wants to investigate drug testing, organic acids, etc. from sweat. And it's also things that people have looked at in the psychiatric area, where sweat and doing metabolomic studies from sweat samples. So Brett would know the very small sweat coils, but the same company produces very big ones to actually do collections, which is specifically for metabolomic studies. So it's there, I think. That's the Westcore system. It's a large coil.
Just a quick question regarding like the vancomycin bridging studies that you've been doing. Was that with a finger prick as well?
Yes, so we did peripheral whole blood VAMs and a peripheral capillary sample. Yeah,
I was just interested in the levels were different, and I was just wondering, say in a septic patient with microcirculatory dysfunction, if you can take a sample from, say, the chest or the tongue or somewhere that is more vascular. Has that been done or investigated previously? Like taking, say, a sample from a central area where it might be better perfused than, say, a digit?
Yeah. I haven't read anything in the literature about it having been done. It certainly would be feasible. I hesitated in putting that data up because it's one patient and there's so little information there. I also Brett mentioned too that when you're taking a sample from a digit, what are you actually collecting? And so I think it would be the same thing. But yeah, definitely, it would be very interesting to see if there's a difference.
So I guess there's some data from the microdialysis literature. I mean, it's looking at interstitial fluid concentrations as opposed to blood concentrations, but there's certainly a big difference in patients with hemodynamic compromise.
There's also some literature around taking earlobe in really young kids, so you can do a earlobe prick, and that may bleed a little bit easier. But obviously you have to twist the head around to get it onto the spot, or the spot around eye.
Okay, so I've got two more questions. The next one relates probably mostly to Brett, but Steve may have some comments and Lauren's maybe as well. But it relates to use of microsampling for clinical care more generally. So not just measurement of drugs, but electrolytes and haematology. What do you think the likely time course of that may be, if at all?
Well, actually, I was thinking that too. When people are with microsampling, we're looking at a drug level. Typically, they may want a full blood count, or they may want an MBA with that. So the issue then becomes is you've got to look at the big general chemistry analyzers and how easy that happens. And you've got to be careful then if hemolysis occurring, because that's going to affect your potassium level. How long is it stable for? Does the glucose go off? So there's all these other variables that are occurring. And so you've got to be very careful about that. The other thing too is how do all these analytes come off the microsampling device, whatever you've used? Are they reproducible? You've got electrolytes, you've got to make sure there's no contamination issues. So there's a broad thing to look at, and I think for people in general chemistry, it would be a real paradigm shift for them. And I think you'll find that for a lot of people in pathology, it would be quite scary for them. I think they want the I's dotted, the T's crossed, and the T's dotted and the I's crossed as well, because patients' lives are dependent upon electrolyte results. So I could see it happening, but not for a while.
I don't know if it is already happening, but I learned a few years ago that there were some apps actually trying to look at these things. So you just pinch your finger and just put the sample in a very micro device and then connect it into your mobile phone, and you could receive all this. But I don't know if it's still happening. But
I think it's happening. But I mean, I'll give you an idea. Some of the large pathology laboratories in Australia are doing 20,000 MBAs a day. So whatever system you come up with is going to have to be highly automated, high throughput, the whole lot. And the clinicians are not going to want longer turnaround times. They either want it the way it is or even quicker. So, I mean, there's a lot of barriers to overcome, I think, to go that way.
Do you think one day the intern will be walking around with the point of care testing machine and they prick the patient by the bedside, get the results on the ward round, do a blood spot for the drug level later at that time as well?
Well, it's going to be very interesting because the regulators are going to come in and say, okay, how is it NATA regulated? Your QC's like? Have you got an external QC program you're involved with? How is it performing? What are your training records? All those things that currently now that we have to, pathology, have to guide by. So
maybe a pathologist on the ward round?
You won't see a pathologist on the ward, no. You might see a hematologist, but not a chemical pathologist.
So it's a really interesting question, Jason. And in the clinical research space, where in resource poor and remote areas, just like the South African example, there's obviously a great push to try and see if we can get more out of those finger prick samples. And so as part of the rheumatic fever study, we've also, the group that we're collaborating with has also validated an ASOT, so an anti-streptococcal titer from the spots as well, with standard samples that they have. And so they're being used as one of the PD outcomes that we're using for the PK study. And I think there's potential for in situations where you don't have a creatinine or available, then actually there are some methods out there which at least might provide you a surrogate for renal function. Whether that really matters for small kids is probably a bit of a moot point. The other thing is that hematocrit out in rural areas is easily sort of at least inferred by having a point of care hemoglobin test. And so there's a very clear linear relationship between hematocrit and hemoglobin and Melanesian kids. We know what that relationship is. And so those are relatively easy to measure. But getting the most out of the spots is a great opportunity.
Can I just check with our video conferencing colleagues if there's any questions out there before I ask a final question or ask the audience here? So my final question is that we've discussed on the panel so far just general microsampling and haven't been specific to a device. But what we heard throughout the day is that there are lots of different devices. They all perform differently. Some perform better in some scenarios than others, and others perform better. So there are advantages and disadvantages of all. Do you think that the reality is that there's not going to be one microsampling device which is what we will be using, but we're going to be using a microsampling drug combination or a microsampling patient drug combination into the future, and that is what is most likely to be where the future of microsampling is that there's going to be individual combinations which suit drugs best?
I'll give it a go. Again, I'm coming from a pathology perspective. One thing you've got to be careful of is that we don't collect so many little micro samples from Joe Blogs that are different types, because I know there's going to be an almighty, I won't say the word, downstream, because people will mix them up a whole lot, the process. If you can keep it onto one matrix or one or two at the tops, it makes it easier. As soon as you get a collection which is very complicated, with multiple tubes, the potential for error gets exponential, and then it's a waste because you've got 5 or 6 tubes, and one's lithium heparin, one's EDTA, SST, the whole lot, and they're spun down, they go into different aliquot tubes. So I think if you can try to keep it as small as possible, you're going to minimise potential of error down the line. And that's from from our perspective, that's what we see.
Susie?
I think from the testing that we've done of microsamples, we've sort of had a look at a range of them. And at this stage, I don't see something that is, the definitive microsample. I do think it's going to have to be, just related to the drug, what suits the best patient. And maybe that's why, it will be in clinical research for longer before it can move into TDM or biochemical environment.
Steve?
Already, without microsamples, we have a range of different sample types. And just with, today we've talked about plasma and measuring stuff in whole blood, measuring stuff in RRTE and urine. So I don't think there's one super sample that fixes it all.
Lawrence, did you have a comment?
One of the sort of practical issues we have is that actually there's such a huge lag between designing so let's just say we put a study together and we design a study and we for an application and then it gets funded and then you put it through ethics. The lag phase from start to finish for us is 2 to 3 years from when we have the idea to when we actually do the first finger prick. It's really long. So, and obviously during that time, as we've seen with all these new devices that have come on just recently, the landscape moves reasonably quickly, and it's that's going to be a bit of a challenge. So we're sort of at the moment, we just put in our ethics application for the Primaquine studies and so on, and they all dependent on the filter paper, the dry blood spot. And yes, if in retrospect, if we'd known perhaps, what we know now, we wouldn't have designed them in that way, but we're sort of committed now and we can't go back and change the ethics. So there'll be a big legacy effect of there's always going to be a lag phase in designing these studies, and probably just have to acknowledge that along the way.
And I guess also cost comes into it as well, is that when designing the studies, if we're doing a lot of collection points and large sample sizes, a test that costs $5 or $6 per kit is actually just going to be completely unrealistic for the type of clinical studies that we do. So we really just need to make a robust method and have something well validated and optimised, and then use it as best as we can, I think, particularly sort of in our setting, if we want something that's translatable across field sites, as long as we have confidence in our assay method, I think we're got a strong point.
Can I push back to you, Jason? You're using the microphone you made?
Yeah, you work routinely in intensive care. Can you, if you could use microsampling now and we had the thing, would you use it?
As long as we were confident in the quality of the results, of course.
Right. Okay. I was just wondering.
So one of the issues in ICU, of course, is the amount of blood which gets used for various tests, and so trying to minimise that's something which is important.
Because I suppose it's not just the cost of the device; it's the cost of collection, the costs of changing processes with how it's going to be sampled, the different equipment to use the stuff. So it's a real knock-on price, and you've got to say, well, okay, we've got to look at the whole price for the whole process from sample collection through to result generation, and what needs to be changed. So that needs to be considered. I think that's going to be one of the bigger barriers, because they'll want to know what the cost per test is for the whole process.
Sorry, just to add to the cost, the range of microsamples that we've seen today, the dried blood spots or dried plasma spots, we get that paper for free. And but the other it goes, but it goes right through to the other end, where some microsamples I think are around $14 or $15 per device. So it is something you'd factor into your decision making.
One last comment from Rhonda.
I actually wouldn't have thought you would have used microsampling as in dried analysis for ICU or emergency, because you actually have a time lag, and that will actually inhibit a number of your results that you are wanting urgently. So I wouldn't have thought it would have been the right approach. I would have thought it would have been wet point of care whole blood or equivalent in
I think it depends on the timeliness that you require it. And so if it's let's say an antibiotic level and you're not going to dose adjust until the next dose is due, then there are certain time issues which are actually in your favour if you were to use a dried format, but of course this could be a liquid as well. Well, on that note, I think we should all thank the panel for an excellent exchange and discussion there. Thank you. And then to thank you all for attending in person in video conference land for this first microsampling symposium on drug dosing studies, which I think has been fascinating, really interesting to share a lot of ideas that we have, and I hope that we've all learned things which we can use to further and improve our further research protocols that we aim to develop in coming months and years. I'd like to thank our sponsors, Shimadzu, Astellas and MSD, for the work they've done with this. Our guests that have flown from afar, who have added a lot of quality to today's proceedings. We're very grateful for all the delegates that have also come from afar. I see people from Sydney and Melbourne and Adelaide as well. So it's wonderful that you could have made the effort. But from me personally, I think that I would really like to acknowledge the work of Susie Parker for the work she's done in getting the symposium up and running, and also our CRE manager, Nerman, who is probably outside. I know he's in there. Okay. For all the organisation he's done as well. It's been fantastic. I think the catering's been first class, and everyone has known what's happening at every particular moment because of his excellent organisation. So thank you, everyone, and I wish you a very safe trip home, but please don't leave without stopping by for a drink and something to eat for the next hour or so. And please give Pascal a high five on the way out for his excellent AV work as well. Thank you, Pascal. Thanks, everyone.